The visual shows a man eating all types of food. Let us see how this food gets digested in different steps. The mouth or the buccal cavity contains the teeth, tongue, and salivary glands. Food is chewed in the mouth, mastication, breaking larger food pieces into smaller pieces. Food is also mixed with saliva secreted by 3 pairs of salivary glands. Saliva moistens food, dissolving soluble food molecules such as glucose. Saliva contains mucin, which lubricates the foods so that it can be swallowed with ease. Saliva contains an enzyme, salivary amylase, or ptyalin, which starts the digestion of starch to maltose. Saliva also contains salts such as sodium hydrogen carbonate, which act as a buffer, providing a slightly alkaline pH in the mouth. Esophagus. By the alternate contraction and relaxation of the smooth muscles lining the esophagus, food goes down the tube. This characteristic movement of food is called peristalsis. From the esophagus, food enters the stomach. Stomach. The stomach is a muscular bag with elastic walls where food eaten during a meal can be stored for processing later by the rest of the gut. When food enters the stomach, starch digestion stops because salivary amylase is denatured by the acid pH in the stomach, and protein digestion begins. The gastric glands lining the stomach wall secrete the following substances. Pepsin, a protease secreted in an inactive form called pepsinogen, catalyzes the hydrolysis of proteins to peptides. Renin, found in young mammals, converts the soluble protein casinogen found in milk into solid casein. Pepsin then digests the casein to produce peptides. Hydrochloric acid changes the pH in the stomach to acidic. This gives an optimum pH for pepsin and renin and kills most of the bacteria ingested with food. HCl also helps in the conversion of inactive pepsinogen to active pepsin. Mucus protects the wall of the stomach from the acid and protein-digesting enzymes. While in the stomach, the food is churned and mixed by peristalsis, resulting in chyme with a liquid consistency. The pyloric sphincter is a circular band of muscle at the lower end of the stomach, which allows the chyme from the stomach to enter the duodenum part of the small intestine. The small intestine is about 7 meters long in humans. It is divided into an upper duodenum and a lower ileum. When the food enters the duodenum, the gallbladder and the pancreas release their secretions into their common pancreatic and bile duct, cystic duct, which opens into the duodenum. Bile is an alkaline green watery fluid which contains inorganic salts, mainly sodium hydrogen carbonate, which neutralize the acidic chyme coming from the stomach. Organic salts, which emulsify lipids, breaking them into small droplets. This facilitates the lipases to act on lipids. Bile has no enzymes. Pancreatic juice. It is an alkaline fluid with a pH of 7 to 8. It contains sodium hydrogen carbonate to neutralize the acidic chyme. Pancreatic amylase, which catalyzes the hydrolysis of starch to maltose. Trypsin, a protease secreted as an inactive molecule trypsinogen, which catalyzes the hydrolysis of proteins to peptides. Pancreatic lipase catalyzes the hydrolysis of lipids to fatty acids and glycerol.